Should be good.
So we're ready to go ahead and start.
Yeah, I think so. I'm just worried that I'm trying to put this up higher so I can reach it more easily, but I think we're okay. It should be stable.
If we need to, I can log into the internet or the Wi-Fi.
Okay. So now we have some guard. It's easier for her to walk away from it. You can either walk.
Okay. We're 11:15. We'll go ahead and get started. This is our 6th bio seminar of the spring 2022 semester. I just think about that for a second. But today we have Dr. Andrew Mehring, an assistant professor in the biological sciences department over at the University of Louisville. And we're going to let Dr. Richter give the sort of introduction. But I'll just first ask if any sort of announcements for the department. Okay, you take it away.
Thanks. I just met Dr. Mehring today in person. I emailed with him before, but he'll be one of our colleagues working in the wetland system of the Daniel Boone that you've heard about, and faculty and students are involved in. So Dr. Mehring is an assistant professor of ecosystem ecology in the biology department, University of Louisville, and is currently recruiting graduate students. So if you're interested in a PhD studying ecosystem function in streams, wetlands, forests, and urban ecosystems, we'll be going to lunch after, as we always do. So, and make sure you come to that. And then after lunch, we'll have a group meeting up in 3119, if anyone's interested in attending that. I'm going to start with your highest achievement, in my opinion, which he's a 3-time winner of the University of Georgia Odom School of Ecology's annual chili cook-off. Yeah, start with that.
Sometimes, yeah.
He's lived and conducted research in Pennsylvania, where he's from, Georgia, California, Australia, Spain, Costa Rica, and the Netherlands. Happy to be in Louisville to call it home now. But he had a Marie Curie fellowship, and it's a underutilized source for U.S. students and faculty postdocs to do work in the EU. And so there's funding out there for it. He was working at the Doñana biological station on the coast of Spain. But if that's something you might be interested in as well, join us for lunch or afterward, and he's be happy to fill you in on it. So thanks for coming today, and we look forward to hearing about your research.
Thanks. Yeah, thanks so much for having me. Thanks a lot. I've been meaning to get out here for a long time. Actually overlapped with Dr. Mott long time ago at Shippensburg University, and also interested in a lot of the work happening at ridgetop wetlands here and in the natural areas around in Daniel Boone National Forest and in this region here. So it's great to be here. Thanks a lot. As far as the chili cook-off, sometimes a creative name actually can win it for you. So, and I can't take credit for this one. It was a professor who won in the spicy chili category one year, and the name sounded like weapons of mass destruction. But if for spicy chili, if you tweak that just a little bit, you can get a really good name. I won't go into detail, but anyway, I'm going to be talking today. I will talk about flamingos a bunch later in the talk, but I'll be talking today about zoogeochemistry, and I'll tell you what that is exactly, and talking about different ways that animals affect ecosystem functions in biogeochemistry. So I am an ecosystem ecologist, and I'm sad to say that the whole field of ecosystem ecology can be summed up with this very simple diagram. Basically, we're interested in looking at whole ecosystems and measuring the movement of material and energy into the ecosystem, through the ecosystem, and back out the other end, or back out the sides. You know, it's hard to draw boundaries around these things sometimes, but that's kind of what we do. It's a simplified version of it. I worked mostly in aquatic ecosystems, wetlands, lakes, streams, and rivers. But I also do some work in forests and other terrestrial ecosystems as well. And I'm interested in how, in biotic effects, mainly how plants and animals are affecting ecosystem function in both aquatic and terrestrial systems. And especially now, I spend a lot of time measuring fluxes of carbon dioxide and methane and different greenhouse gases like nitrous oxide as well. And I'll talk a little bit about why I'm interested in those things. So I measure carbon flux, and there's lots of good reasons to do that. There's lots of things you can learn just by measuring the movement of carbon dioxide, for example. Like if I were to put a clear chamber like this one that allows sunlight to pass through, but it's gas tight, and I can put this over a plant, and measure the rate at which carbon dioxide levels are decreasing, I can actually measure the rate at which plants are pulling carbon dioxide out of the atmosphere or sequestering carbon. Similarly, I can take one of these floating flux chambers and put it onto the surface of a wetland and measure the amount of CO2 crossing the water surface into the air, or methane crossing the water surface into the air, and I'm getting measures of greenhouse gas emissions from wetlands. Or, and something I'm just starting to teach myself now, this is going to take a while because it gets more complicated, but you can put a sensor on a tower mounted above a whole ecosystem, and you can actually measure emissions or even uptake of greenhouse gases like carbon dioxide from a whole ecosystem. So you can learn a lot by tracing the movement of carbon. Now, humans store carbon. I know you don't often think about it that way, right? We hear about humans and greenhouse gas emissions, and that's what we hear about most of the time. We're increasing the amounts of carbon in the atmosphere, and that's happening. But in your body, you are actually storing carbon. If you think about it, you started out as a cluster of cells, and you certainly now contain more carbon within your body, which actually physically in you, than you did when you were, 1 year old, 5 years old. How much? Well, if you want to know how much carbon you have accumulated, you can just basically take your weight. We're about 18% carbon, roughly, give or take. Take your weight, you can divide it by 5. That's going to be a little bit off, but it's close. And that's about how much carbon you contain. Now, where did that carbon come from? Well, obviously, it's from the food that you ate, right? We don't photosynthesize. We can't just get carbon from the atmosphere. Now, whether or not you ate, you're eating meat, or you're a vegetarian, vegan, whatever you're eating, at some point, the food that you're eating probably was eating a plant, right? Now, where do those plants get they get the carbon from the atmosphere, right? They're taking up CO2. Where did that carbon come from? Obviously, we talk about greenhouse gas emissions, but where else does carbon and the CO2 in the atmosphere come from? Does anybody have a hazard a guess? Respiration, right? Yeah, respiration. Sometimes decomposition, right? So microbes, for example, are respiring, and we are releasing carbon dioxide. This room is going to have a higher carbon dioxide concentration by the time we leave than it did when we came in, I guarantee. Bacteria and things are also going to be releasing carbon dioxide, just like we do. When things are decomposing, the bacteria and fungi are also respiring, giving off CO2. So that CO2 that those plants were using may have at one time been part of some other animal or some other plant that broke down. When we all eat these foods, the carbon in those foods actually becomes incorporated into our bodies. So you might have, if you ate a burger yesterday, you might have an atom of carbon that was once basically the same atom of carbon that was inside of a cow, right? These things remain carbon atoms, but they just get passed around between plants and animals and microbes, and that carbon atom just keeps cycling over and over and gets passed from one organism to another, one place in the ecosystem to another. So another way of thinking about this, if you want to go a little bit further back, is imagine that maybe a katydid was eating some leaves, and then it died next to a plant, and then the katydid starts to decompose. The plant leaves take up the carbon that was once in the katydid because the microbes break the katydid down. And now that plant contains some carbon from a katydid. You eat the plant, you now get the carbon that was once in the katydid, right? I mean, and I think this is stuff that you learn in basic biology classes, but you don't always think about it this way. You can go even further back and say, there's a pretty good probability that a lot of you in this room actually contain carbon atoms that were at one time inside of a dinosaur. Or your food might have carbon atoms that were at one time inside a dinosaur. And with even more certainty, you can say that a lot of carbon in our bodies was at one time part of a star. So it's kind of neat stuff to think about. Now, at some point, you're going to lose all this carbon that you've accumulated. I know it's kind of heavy to think about. You probably would rather talk about chili cook-offs, but the thing is that carbon, once is released to the ecosystem again, will become part of something else. A coral reef, a sea turtle, maybe giant sequoias, maybe even another person. So this stuff just keeps cycling. And this is kind of what ecosystem ecologists do. I try to put it into a perspective of thinking about what it means with humans, but we trace the movement of elements through ecosystems, just like you can trace the movement of elements between the food that you eat and other things. Well, what else is kind of cool about this is you don't even have to wait until, the distant future to donate your carbon to other organisms, because your body is replacing carbon all the time. Lining of your stomach, about a week, a lot of those cells have actually been replaced. And when you exhale, some of that CO2 that you're breathing out a lot of it's because of the breakdown of cellular respiration. Some of it, though, when you break down fats, you actually breathe it out. So something that might have been physically at one time part of your body, you exhale it, and that could get taken up by plants. So there's a good chance if you have houseplants, they might contain carbon atoms that were also a part of you at one time. And especially if you talk to your plants, there's an even better chance of that happening. Anyway, and I talked about humans storing carbon, but of course, there are a lot of other things that are very much the opposite. We take carbon from deep underground, we mine it, and we burn it, and then release it to the atmosphere. And of course, that contributes to things like the warming of the land and oceans. And that's an effect that we have on ecosystem function as well. And that's basically where zoogeochemistry comes in, because we are animals. So humans alter carbon cycling, and lots of other animals do as well. So biogeochemistry, it's actually, when I was at Shippensburg, I first heard this term. It was a visiting partner of one of the other grad students. He told us he was a biogeochemist. And at the time I was a master's student, I thought that sounds terrible, because I know chemistry was hard. And then there's also geology and biology, and it's all in one field. And that sounds really, that sounds rough, but that's what I ended up doing now. So this is basically studying the physical, chemical, biological, and geological processes that govern ecosystem structure, like the community of organisms, and also the way the ecosystem functions. Zoogeochemistry is the role of animals in biogeochemistry. So a couple of examples, things that you all have probably heard about. Salmon, for example, they hatch out as eggs in headwater streams. A lot of them in the Pacific Northwest. We used to have a lot more salmon on the East Coast as well, Atlantic salmon. Hatch out as eggs in freshwater streams. They feed on freshwater invertebrates for a while. And then once after maybe 2 or 3 years, give or take, they move out to the oceans, and they spend years in the ocean as predators, feeding on smaller fish. And then before the end of their life cycle, they come back to these headwater streams. They move upstream in these mass movements, and then they spawn and they die. And after they die, of course, you see bears, and even catching them before they die. Bears are catching these salmon as they're coming upriver, right? And wolves are maybe getting the carcasses and dragging them off into the woods. So you basically had this massive movement of carbon, nitrogen, phosphorus, other elements from the ocean, basically that had accumulated for years in the ocean, all of a sudden being brought back to the land. Right, so this is a big transport of elements that's being mediated by animals. There's been some cool research around this. For example, some researchers here studying trees, taking tree cores. I think you had Megan Rockner here giving a seminar, so you've probably heard about this before if you attended her talk. Taking tree cores and actually measuring the isotopic composition of elements within these trees. You can actually, by looking at the different isotopes of nitrogen, you can measure a signal of something that might have come from an organism from another ecosystem. And they're tracing, basically looking for signs of nitrogen that came from salmon. And so the salmon swimming upstream, getting dragged into the forest, dying, they could actually see down here, you're looking at this isotopic nitrogen, and this elevated signature is because of salmon. Is in this red line. These are actually trees that were in streams that had high numbers of salmon swimming upstream. And they could actually measure the signal of this huge movement of salmon, all this nitrogen being incorporated into the surrounding forest around these streams and rivers. And not only that, they also could see these signatures of the salmon, the nitrogen from the salmon, and predatory ground beetles, these predatory beetles in the forest floor that were basically eating the other invertebrates that were scavenging the salmon carcasses. They saw a signature of the salmon nitrogen within all, things like huckleberry, salmonberry, shrubs and things in the riparian zone. So salmon were bringing this huge influx of nutrients into the ecosystem, fertilizing the ecosystem. And then of course, not just as moving things, but also storing carbon. Something that you have in this area, beavers, they build dams, right? Dam up streams and rivers, slow down the flow of water. And that causes lots of organic matter to drop out of suspension and settle into these ponds. And over time, this is looking at megagrams of carbon. This is some research from Ellen Wohl. And this is looking at megagrams of carbon, and everything below the 0 line is carbon stored underground. This bar all the way to the right is an abandoned beaver meadow. These beaver wetlands were massive sites of carbon storage. And it was estimated that at one time, the population of beavers in North America was somewhere around 500 million. That's more than the population of people in the United States. Now it's been drastically reduced. And at one time, because of trapping and things, the numbers of beavers got really, really low. And it's estimated that when that happened, we lost a lot of carbon storage in North America. So these animals are actually contributing to storing carbon underground by creating these dams. And then also studying, measuring fluxes. And this is a project where I was collaborating with some other people. That's Rose Martin there. We were measuring some fluxes of gases from leafcutter ant mounds. And similar to our landfills, leafcutter ants, how many people have heard about them? Okay, so you've heard about leafcutter ants, right? They go out into the forest, they cut leaves, bring it back to their nest to feed a fungus, right? And then once that leaf material is spent, they take those leaf particles and take it to a refuse dump that's also, depending on the species of leafcutter ant, probably going to be underground somewhere. It's much like our landfills. We bury organic matter and trash in landfills. And because that organic matter is decomposing in an environment with very little oxygen, instead of being converted to CO2, a lot of that carbon, a lot of that organic matter gets converted into methane. And so we're putting flux chambers over these openings of these leafcutter ant nests, and we are finding these massive emissions of methane from these leafcutter ant nests, just like you would expect from a landfill. So, organisms, again, affecting the ecological ecosystem function in these rainforests. Oh, and that's just a figure showing, this is basically normal soil flux. Methane's right around 0, or even might measure uptake because there's microbes that actually consume methane in soils. In the leafcutter ant nests, this is going from 0 to -2 up to +2. This is going from 0 up to about 15,000 milligrams of carbon per square meter per day. So a huge increase in methane flux, methane emissions. But what I want to focus on today is talking about benthic invertebrates, things that you find in wetlands, and how they enhance greenhouse gas emissions. There's a couple of different ways that this can happen. These things that you see over here are tubificids. I think now they're in the family Cnidids, but these are oligochaetes. I know you have a bunch of them. I was talking with Brady about you have a lot of these in your wetlands around here. These worms burrow head down into sediments, and they usually feed down these deeper layers, and then they respire up in these upper layers, up above the water column if the oxygen levels are low. These lower layers of sediment down here are the anoxic layers. There's not a lot of oxygen down here. This is where methane is getting produced. And so these worms, by creating burrows that reach down to the anoxic zone of sediment, might make it easier for methane to escape into the overlying waters. They also are called conveyor belt feeders because they feed head down. They're always removing the lower layers. Organic matter is passing up through the worm, they're pooping it out up above the sediment surface. And so there's basically this constant cycling of sediments, which might move really nutritious organic matter down to the deeper layers of sediment at a faster rate, make it accessible for the microbes that produce methane than if there was no worm present, right? So that could potentially enhance methane that way as well. Over here, I don't know if anybody recognizes this little fly larvae. This is a chironomid, right? This is a non-biting midge. They build these U-shaped burrows, and there's been some cool research out a couple papers down here. This is a growing body of literature. But these fly larvae will undulate their bodies and create a pool in fresh water and also nitrate into their burrows. A lot of that nitrate, because it's a very short travel time through this burrow and also through the gut of the chironomid, which is anoxic as well, that nitrate then gets converted into nitrous oxide, which is another potent greenhouse gas. It's basically incomplete denitrification because this nitrate gets pulled into this anoxic environment, and it's not in that environment long enough for denitrification to proceed to completion. So you get a lot of this nitrous oxide, which is sort of an intermediate product of the denitrification process, and not what you really want as far as greenhouse gases go. So a couple of different ways that invertebrates have been shown to enhance greenhouse gas emissions. One that gave us this inspiration for this research I'm going to talk about today. I teamed up with a researcher who works with water birds in southern Spain. And he's done some experiments before with other co-authors where they're actually looking at the effects of things like flamingos and ibis on aquatic invertebrates and also on plants. What you're seeing here is a wetland in Spain, and this is the shadow of a plane that's flying over. They do monthly water bird censuses here. It's really a high level of high bird diversity. And just before this plane flew over, this wetland was full of hundreds of flamingos. They make these little circular structures that you can see here, and during their feeding. And the way that they do that is actually, there's something like this video will play. It's like this. They do this little dance. They're basically stirring up a cloud of sediments, and they're getting a mouthful of sediments and invertebrates in water. And they have, inside of their bills, they have these structures that are kind of like baleen in a whale. They're called lamellae. I can actually show you here. It looks like this. When they close their bill, it's almost like baleen. They can basically get a mouthful of stuff, These structures are perfectly spaced to capture the particle sizes, usually the acronymids, worms, and things that they want to eat, and let the sediments and water pass back out into the environment. And they're actually really efficient filter feeders. So they actually can reduce the number of these invertebrates living in sediments. They also sometimes, because of the stirring up of sediments and things, they can reduce plant biomass as well. So this project, this Marie Curie project, was called Flamingos, and it's obviously misspelled. Actually, I was giving a talk on this and somebody corrected my spelling once in the title. But that stands for Functional Links in Avian, Macrophyte, Microbial, and Invertebrate Greenhouse Gas Output Stimulation. And we were interested in the effects. I think that also helps when you have a catchy title or an acronym for a project. But we were interested in these flamingos and how they were affecting invertebrate densities in sediments. And then the reason we thought that might have an effect on greenhouse gas emissions is that if you have more birds, more predatory birds like flamingos and ibis, which is actually happening in the southern coast of Spain and France and some of these wetlands, they're increasing over time, then you might have fewer invertebrates because they can reduce the number of invertebrates in these sediments. And if invertebrates were enhancing greenhouse gas emissions, well, with fewer invertebrates, you might also have reduced microbial activity and less greenhouse gases. So that was the idea we were going with for this. So there are two components of this project I'm going to briefly talk about here. One was a lab study, which was looking at the effects of invertebrates in sinking algae on greenhouse gas emissions. And then we have a manipulative field study, which was looking at the top-down effects of water birds on greenhouse gas emissions. When I talk about top-down effects, have you all learned about that in your classes? Okay, so like, yeah, cool. So predators basically having an effect on prey that then has an effect maybe on lower trophic levels potentially. So this first laboratory study, during these Marie Curie projects, you're usually required to spend about 3 months at another institution, not your primary institution, but you have to go somewhere else. So I actually went to Wageningen University in the Netherlands. That's my partner Meredith over there. She was along for the ride, and she was helping me out in the lab that day. She was actually teaching English at the same time. People don't realize how easy it is to get a job teaching English for a short period of time in the EU, where someone actually pays you while you're there. You also get health insurance coverage and all that stuff. There's lots of things that people I think aren't aware of that I'm happy to share information on. But she was teaching English while we were over there. Anyway, in this experiment, we were using these oligochaetes, and we had them basically in these mesocosms that you can see over here, in a temperature-controlled giant water bath, right? And so we had these tubificids, which are known to potentially enhance greenhouse gas emissions, and we had different densities of worms. 0, 15,000 per square meter, and 30,000 per square meter. Those are realistic numbers. Those are actually pretty conservative because these can be in really high densities. If you ever picked up a sample, you might have seen them. And then we crossed that with another treatment, which was dead green algae. And we had this representing conditions you might have in wetlands that are getting different amounts of nutrient loading, oligotrophic, mesotrophic, and eutrophic conditions. And this was about 7 or 25 or 63 grams of carbon per square meter per year. And this is to represent the amount of algae you would expect to be sinking in a wetland that had algal blooms, right? So we were using literature values and then trying to add, it was chlorella from the Milton shop, which I think means the nut shop. You can put it in your smoothie, but we used it because it's a nice green alga that's very easy to consume for invertebrates. So we're adding this with the idea that with this labile, the source of carbon, we would see high methane fluxes at the highest levels of worms and the highest levels of algae. So those two things combined, we should see really high methane fluxes, because the worms, if you remember when I was talking earlier, are going to mix that algae into the sediments, and then that's going to feed the microbes that produce methane. We also thought that this effect might depend on oxygen levels because you need low oxygen in order for methane to be produced. So we had half of these mesocosms aerated with bubblers and the other half not. And you can see the results here. This is from the end of the experiment, it ran for about 6 weeks, and this is a methane flux. And this is methane flux, and this is grams of carbon, methane carbon, per square meter per hour on the y-axis, going from about 0 to 0.1. And we have our tube effect worm density. We bought the worms at a pet shop, which is great. You can get bags and bags of them to add to mesocosms. People usually use them for feeding their pets, but so this goes from 0 to 15,000 to 30,000. And you can see over here that, oh, and I'm sorry, I should point out also that the red is low algal concentrations. The green is medium algal concentrations, and blue is high. And so you can see that when we had levels of high algae in blue and high density of worms, we saw high methane emissions, and there was a significant effect. So up here is this R squared 0.86, about 86% of the variation explained by an interaction between worms, algae, and aeration. And that aeration was very important because when you aerate the system, when there's lots of oxygen available, there's basically no noticeable effect of worms or algae here because it's oxygenated, so methane is not going to be produced very easily. So methane flux was highest at the highest levels of worms and chlorella, like we predicted, and methane flux was lower in oxygenated mesocosms. Now for carbon dioxide, also looking at the results from the end of the experiment, it wasn't exactly the same. Again, this is CO2, grams of carbon per square meter per hour being emitted from these mesocosms. And I forgot to mention, we actually took a portable greenhouse gas analyzer to measure these emissions and you just fit it right down on top of each mesocosm. So we could do about 36 mesocosms in a day, getting a nice flux of CO2 and methane from these little treatments. And here again, you can see with highest levels of tube effects and algae, basically over here to the right. In blue, we saw the highest emissions of CO2, but it seemed to be really dependent on algae more than, when algae levels are high, you always have lots of CO2 being emitted. The other thing that was different here is that, we saw CO2 flux highest at highest levels of worms and algae, but there was no effective oxygenation. So the CO2 just being produced and being emitted at roughly the same rates at different levels of treatment, whether there's oxygen present in the system or not. All right, so invertebrate enhancement of greenhouse gas flux was influenced by the availability. So the invertebrates were enhancing greenhouse gas flux for one, but it was also influenced by the availability of labile organic matter, sinking algae. And the interactive effects on methane flux might be limited in oxygenated environments. Now, for the manipulative field study, we're looking at the top-down effects of water birds on greenhouse gas emissions. This is basically what we were doing. It's a pretty simple design. To exclude flamingos, flamingos can live to be very old. I think the oldest one known, this is greater flamingo, was 85 years old. This is one that was 85 years old in captivity. Long-lived birds, I'm not sure how smart they are because you can keep them out of an area with 4 posts and a stainless steel cable about 70 centimeters off the bottom of the wetland. And I would watch them in binoculars and think, you could duck under there, but they didn't. And we would observe these music, they would just get to the cable and turn around, probably because these are huge wetlands, there's plenty of space to go around and why bother stooping under something if you don't have to. But we had these treatments that were keeping these flamingos out, and we compared them against a control which had no cable, and the flamingos would happily walk into there and feed. And the hypotheses were that invertebrate densities would be higher inside the exclosures, and as a result, fluxes of CO2, methane, and nitrous oxide will also be higher inside the exclosures. So this was done in 11 different wetlands. This is along the coast of Spain. This is, here you're seeing these, if you can make these little lines out, these are sand dunes in Doñana National Park. And we're working from basically the city of Cadiz along the coast all the way up to ODL and in different wetlands, and wetlands of different types as well. They had some places that were hypersaline. This is where they make sea salt. I have a new perspective on sea salt because I realize now that the places that make sea salt are not out in the ocean. They're in these estuaries and birds love them. And the birds are contributing to the sea salt because they feed and poop in the places that make sea salt. And I'm not sure how they get the poop out. Maybe I don't want to know. But they're hypersaline, 130 parts per thousand. Sometimes we saw 160 parts per thousand. That's 4 times as salty as the ocean. So when you came out of the field work, you were all crystallized and crusty with salt. Also a sustainable fish farm that had a salinity just slightly below ocean levels, 20 to 30 parts per thousand, where the ocean is about 34 to 36. And then we also had these natural freshwater wetlands within the park, which were the hardest places to work in because the weather doesn't cooperate. As anyone who does field work in wetlands knows, water levels fluctuate a lot, and this is what it looks like during certain times of the year. You can see some horse tracks out across. There's barely any layer of water left anymore. And we had a really hard time working in these systems and actually collecting data in these systems because of the limited availability of water. They dried out completely. And then basically what we did was we measured water to air fluxes with this floating flux chamber and a portable greenhouse gas analyzer. We measured CO2 and methane in the field. After we're done, we would jump in, get some sediment cores, and measure fluxes of CO2, nitrous oxide, and methane in those cores back in the lab by connecting it to a laboratory analyzer for greenhouse gases. And you can even see the tunnels of some invertebrates in some of these cores over here, in these sediment cores. Sediments were sampled for invertebrates after the incubations were done and the gases were measured. Also, we measured the chemistry of the sediments as well. For the water column, we looked at chlorophyll, total and dissolved nitrogen and phosphorus, dissolved organic carbon, dissolved oxygen, temperature, pH, and salinity. And here are the results. So in these plots I'm going to show you here, we're basically looking at the effect of the exclosure where flamingos were kept out, and I'm subtracting the control from all of these where the flamingos had access. So basically we're looking at the relative flamingo effect, right? This is the difference in those treatments. And this is looking at the difference in invertebrate densities from about 0 to 50,000. This is per square meter, and this is exposure minus control. So anything that's above this 0 line means there were more flamingos within there were more invertebrates when flamingos didn't have access to a section of wetland. And this was actually run on log transform data, and you can see that in the inset box up here. But there was a significant effect on all bioturbating, things that were tunneling in the sediments, invertebrates, which are things like the polychaete, burrowing amphipod, oligochaete, and chironomids. We had a chironomid that lived in salinities of 160 parts per thousand. Chironomus salinarius, appropriate name. So yeah, there was a significant effect on these groups of invertebrates, whereas there were higher numbers of them when flamingos were absent. As far as what we're really interested in is greenhouse gases. With flamingo exclusion, again, we're looking at the flamingo effect. This is milligrams of carbon per square meter per day. There was a significant enhancement of CO2 emissions from the sediment to the water when flamingos were kept out. You can see that here. And it was an increase of about 1 gram of carbon per square meter per day when flamingos were kept out. So significantly higher CO2 emissions. And that's actually a pretty big change on a daily basis. Now, not entirely sure if that's because there were lower numbers of invertebrates, which would be respiring, like we talked about earlier. They're going to exhale CO2 just like we do. Or if it's because the flamingos were kept out and they're not disturbing the sediments anymore, and that's changing the biofilms. We didn't see the same effect. We saw changes in the same direction, slightly elevated CO2, but not statistically significant. We looked at the emissions from the water to the air. So there might have been a lot of uptake by algae of CO2 before it actually escaped the wetland. And this is basically looking at what other factors explain carbon dioxide flux. It was related to, these are all different plots showing the effect of these different parameters on benthic flux of carbon dioxide in milligrams per meter squared per day, basically being affected by things you might expect. We have temperature on the x-axis here. As temperatures increase, you see higher CO2 emissions, increased cellular activity, right? If we add nitrate and nitrite, we actually saw decreased CO2 emissions. If you have higher concentrations of those two nitrogen compounds, invertebrate density overall caused an increase in CO2 emission, and also the amount of sediment and the sediment concentrations of nitrogen, if that got higher, that also increased emissions. And then we had this term that if there was an exposure in that area, we also saw higher emissions. And what that says to me is that there is something else about keeping flamingos out that's an effect that is beyond just what the invertebrates are doing, because we are using something called EKU's information criterion, which is a way of selecting among a bunch of different competing models. And it always wanted, basically among all the models we're comparing, it always wanted to keep a term that says the exposure matters. And if that was kept in and all these other things were accounted for, it means there's something beyond just the invertebrates that might be having an effect here. But we don't know what that is yet. For methane, we got some really good relationships here with a similar plots to what I just showed you on the last one. As salinity increases, we see a really sharp decline in methane production. So methane emissions going down steeply as you get higher salinity. An increase in methane production with nitrate and nitrite. Again, invertebrates, the more of them that are present, the more methane emissions you get. And just sediment nitrogen concentration increases, you also see an increase in methane emissions. So in conclusion, invertebrate densities were higher in the exposures, especially for large chironomids, oligochaetes, and polychaetes. Greenhouse gas flux was higher in the exposures. It was true just for CO2. I wish we would have seen enhanced emissions of all three. Nitrous oxide and methane, though, were not significantly affected by the flamingos being kept out. And the emissions actually from the surface to the atmosphere were not significantly different. This was just the movement of gases from the sediment into the water. So we saw this enhanced CO2 flux without flamingos might be due to the higher benthic invertebrate densities and their respiration, but it also could be due to benthic biofilm communities. And we actually sent samples to some researchers at the University of Valencia and the University of Granada who are actually doing molecular analyses now to see if the benthic community of microbes and algae changed. That extra exposure term in the CO2 flux model suggests that there is something else going on. Maybe the biofilm structure or the structure of the sediments has changed. But we didn't actually see any significant differences in sediment carbon and nitrogen content. So it's not like there were significantly more biofilms or more bacterial biomass or algal biomass, but maybe something else about the community players is different. And then interestingly enough, greenhouse gas emissions were highest in the freshwater sites, and this was especially during periods of low water. And a lot of that is potentially because of this concentrating effect of all the wildlife being pushed into smaller and smaller aquatic habitats. And as they do that, they're also, pooping in a smaller and smaller volume of water, which is going to further enhance emissions, which is, the guano effect is certainly important as well. And there are legacy farmers out there who still have grazing rights, and so these wetlands are also full of cows in some cases, which are contributing some carbon. So, do I have time? I could breeze through this. This is just talking about. Okay, great. Okay, and that still leaves time for questions. Okay, cool. Just talking about what I'm doing now. That work in Spain is finished. Love to be able to keep collaborating with those people, but right now I'm focusing on getting some research going locally. Working with the grad student Mark Tierney and Dr. Andrea Gahn, who's in our Department of Environmental and Geographical Sciences. They've changed the name. It used to be geography. And we're actually looking at greenhouse gas emissions from wetlands and trying to see if we can detect potential problem areas with remote sensing. There's a satellite that flies over this area every 5 days. And it can actually measure levels of greenness coming back off the landscape. How many bodies of water do you all see over in this picture on the right? Yeah, there's 4. And some of them are hard to pick out, right? They look like a golf course green. Yeah, there's some very, very green bodies of water here. And oftentimes the more algae you have in a body of water, that algae is really easy for microbes to break down. Like we talked about the experiment earlier where you added the dried algae. That might enhance methane emissions. And there are sometimes correlations between chlorophyll concentration in the body of water and the amount of methane being emitted. So we're trying to see if we can detect that. We have to go out and try to get samples as the satellite is flying over, and we're not getting great sunny days lately, unfortunately, but so it's challenging, but it's really fun. I'm also measuring emissions from these wetlands as well. I'm also conducting some research not far from here and collaborating with Megan Rockner and Stevie Noviak at University of Louisville. And we're working in Berea Forest, looking at the effects of fire on forest carbon budgets and also hopefully on methane flux. If I can get a graduate student to start collaborating on this. Right now, I'm sampling these leaf litter collectors myself. It's not so bad because it's very pretty in Berea Forest, especially during the fall. And comparing burned and unburned sections of forest. And you can actually sometimes see the difference. If you look, this is the side that was burned, and you notice all of those understory trees, the leaves are gone. And we're actually starting to see an effect of the fires on the amount of leaf litter coming down in these different plots of forest. About half the amount of leaf litter coming from the forest canopy back to the forest floor in burned versus unburned sections of forest. And the hike is not so bad, so I don't mind it. But that's my route in the 2 areas between litter fall collectors. And I'm about to install 20 more this summer, and that starts to grow. So at some point, it becomes unsustainable for me to do it myself. But I don't mind being out there. I'll be there tomorrow, actually. And then working with a high school student right now on tree methane emissions. Trees actually emit methane, and we're interested in how people's land care practices in the urban environment might change the amount of methane coming out of tree trunks. The reason trees emit methane is that the roots are tapping into those deeper layers of soil where methane gets produced. That actually allows the gas to escape the microbes that would otherwise consume it, and then it leaches out through the trunks. If people add a bunch of water to their lawns, it's probably making the oxygen levels in the soil even lower. And we also have this really intense urban heat island in Louisville. And as temperatures rise, methane emissions might rise as well. And then the other thing that I'm really interested in talking with people here about is my first research experiences were in forest ponds, and I would love to collaborate with people here on looking at things like the effects of forest composition on pond carbon cycling, or how invertebrates in these ponds affect greenhouse gas emissions. And also potentially, because I know you have them here, looking at some of these beaver wetlands and how they affect ecosystem restoration and carbon storage. Potentially collaborating with some engineers at University of Louisville on some of those questions. That's it. Hope I didn't go too long. We're good? Okay, so let's just have to acknowledge tons and tons of people, because this stuff didn't happen without a lot of help. People even helping me get unstuck in mud in some places. But yeah, lots of people to thank, as well as the European Commission's Horizon 2020 program. And if anybody has any questions, I'm happy to take them.
So again, well, as usual, we'll sort of just start with the in-person questions and we'll move on to the Zoom questions. So, how do I know you have a question?
I always have questions. So I have two questions actually. One, so you said that, or you showed that CO2 emissions increased when you didn't have flamingos, but that methane didn't. Did you see that methane decreased? Because if I understand correctly, methane needs to be formed in an anoxic environment. Presumably those sediments are anoxic. And so could flamingos walking around in those anoxic environments actually release methane? I guess like you can have a balancing effect where CO2 increases without flamingos, but methane decreases.
Yeah, no, that's a really good question. We were thinking along those same lines, like, that maybe, if anything, I mean, there was no significant difference in the methane fluxes, but it was slightly skewed towards being maybe a little negative. Like they might have reduced, when flamingos are present, they might have reduced it a little bit. I hope I'm not telling you that backwards. But the mechanisms that you talk about are make total sense, right? A flamingo goes through, methane bubbles get trapped under those surface layers of sediments, and they can sometimes get released suddenly as bubbles. And the flamingo walking through there would do that really effectively. The other thing is the flamingo stirring it up might oxygenate the sediments, which I think you might have mentioned. And then that's going to make it harder for those the methane producing microbes to actually make methane. So it totally makes sense. I'm not sure that we saw it, but we're actually going to analyze the sediments to see if there were differences in the microbial communities, because it could be totally related to disturbance like we talked about. Yeah, good question.
The other question is just that if you know about research in this topic, I'm really interested in invasive species. Is there research on nutrient flux in relation to, say, invasive plant composition or even like invasive herbivore presence absence?
Yes, plants have huge impacts on methane fluxes in wetlands because they're actually similar to what we talked about with trees. They act like straws. So the plant is tapped with roots down into the layers of sediment, and then the methane gets into the roots, bypasses the layers of sediment where microbes might consume it, and then it goes up through the plant and gets to the atmosphere. So an invasive plant could have that effect. Natural plant would also have that effect. Animals, yes, it depends on what kind of invasive animal you're looking at, but there's been some herbivorous birds that actually clip the tops of plants. They go in and they're herbivores and they're feeding on the plants, and that can change methane emissions as well. Fish are a big one. Invasive catfish, because they go tunneling through sediments and things, can change the structure of the sediment and maybe release gases as well.
Thank you.
Sure.
So using remote sensing to classify them in chlorophyll in pond, I assume sample at the same time. Satellite point.
Trying to get out there right when the satellite's going over if we can.
How are you going to classify the color of green based off the satellite?
The satellite actually can detect the light and the wavelengths that are in the green wavelengths, yeah. And so they have, based on, and the Sentinel satellite, I think has, I can't remember how many different bands of wavelengths, but it's many different. Did you say 7? Okay, so you know this probably better than I do. So right now, the reason I work with Andrea Gahn is she is a remote sensing expert, but they have different vegetation indices based on those, sets of wavelengths, which you can use for calculating, potentially, I guess, NDVI, vegetation index, some other indices as well. But I'm not an expert in that field. So and this would be a great question for me to pass along to Andrea. But do you actually work with this?
Not with but I work with remote sensing.
Very cool. Yeah, I'm really interested too in what other tools are out there. Like we like Sentinel-2 because it has this 5-day return time, and it has a pretty small spatial resolution. But I think there might be some things that are even better now. So if you have some suggestions or anything you want to talk about, I'd love to hear about it.
Any questions?
Do you expect to see the same kind of results with other winged birds? Because obviously flamingos have that lamella that really allows them to get those smaller invertebrates. Do you think you see similar effect with species that maybe target larger prey items?
Yeah, I think, ibis potentially are going in and they have long beaks. They don't really feed the same way. They kind of like just poke in and grab stuff and they pull bigger stuff out. We had a hard time. Originally, we had a treatment that was supposed to exclude all birds, not just flamingos. And it was these floating nets. And these systems were all hypertrophic because of all the cattle, because of actually some poor management of these ecosystems. illegal water withdrawals, drawing the water levels down, things. So everything was super loaded with nutrients. We would try to put these nets out, these floating nets that would keep out all birds, keep out ibis, and they were accumulating so much algae that they would just get bogged down. Nets would break, so they're getting sagging into the water. And I was worried that we were actually getting so much algal growth in these floating nets that it was just going to be adding all these subsidies to the sediments of just algae constantly sloughing off, that I scrapped the treatment. We actually saw higher invertebrates in those treatments, but I felt like this is not a bird effect anymore. And so I can't really answer your question based on the data we had. But I mean, it's a very different mode of feeding. And by removing bigger things, it might have totally different effects. So it's a great question. I wish we could have addressed it better, but we mostly could only really exclude flamingos what we were doing. That's a good question though.
I have more questions, I'll be emailing to you.
Okay.
But two real quick ones. So you're doing your little jar experiments in a lab, and then in your field stuff, you've got much bigger bodies of water than like a little ephemeral pond or something. With your flux analyzer, if you're trying to do a certain amount of ponds in a day, what kind of confounding influence is there on just like increases in productivity as the day gets warmer and things along those lines?
Yeah, no, that's really important. It's a good question because, if you are interested in going out to a wetland and measuring uptake of carbon dioxide, if you want to get the maximum rate of uptake, you got to be out there when the sun is directly overhead. If you measure it late in the day, you're totally missing that effect. So, the timing of measurements matters for things like that. The timing of measurements even matters for methane because it changes in temperature, changes in oxygenation because the oxygen levels change. In a lot of these ponds, they can sometimes shift from being oxygenated to anoxic at night, right? So to get a really complete picture, you either have to do one of two things. Pick a time, do the same time every day. Or if you want a complete picture, you hang out there all night, which I don't really want to do, and measure fluxes. But I mean, yeah, the timing matters big time. Yeah, it's a good question.
The other question is kind of jumping off of Ben's question. Is a lot of what you're doing, it seems like a lot of the other studies you cited are mainly focusing on predation effects. Are there people that are doing similar stuff with things like competition or predation risk without predation kind of getting into more indirect ecological interactions?
Yeah, I mean, it gets really complicated. These data are really messy. Like we sampled so much to be able to get these results, which are not even the cleanest, things you might want to see. The laboratory stuff worked pretty well. We controlled conditions really well, but in the field, it's hard. To be able to show effects of competition, I think there's less of that. I bet it would be really, really difficult, but if you could pull it off, it'd be really amazing. So yeah, it'd be really cool.
Thanks.
I want to add one quick thing. I talked about eddy covariance with these towers, and the beauty of those is that you can install a tower. It has a sensor, and like I said, I'm just starting to learn these techniques. It's going to probably take me at least a year, probably longer. But you can have a sensor out 24 hours a day, measuring the system constantly. Then you come back and download the data, and that's great. And then the other nice thing about that is you have no chamber effects. You're just getting the gases wafting off the system, and it's all natural. And so that's like, for me, like for an ecosystem ecologist, that is like where you want to be. Yeah, so not there yet.
So last call. We're good. Thank you again, Dr. Mehring.